All right, hey everybody. So what we're going to be looking at today as a continuation of our protein synthesis is we're going to be looking at what would happen if the genetic code has some sort of mistake in our sequence. So what we're really focusing on is today are our mutations. So I'm going to go through the PowerPoint presentation with you, and then we'll do a couple sample problems together up here. All right, so here we're looking at our screen and we're focusing on our mutations. So first I want to look at what we mean by protein synthesis, an overview of that, and then we'll get into our mutations. So as a review, protein synthesis is when we're going from our DNA sequence, which is found in the nucleus of our cell. And once again, the DNA cannot leave the nucleus. So transcription has to occur and really transcribe that DNA into an RNA sequence. To go from the DNA to the RNA, we're still in the nucleus, but we need the help from RNA polymerase to make our RNA sequence. Once we have our RNA, we can then leave the nucleus, and we can then go into the cytoplasm so that we can perform translation. We want to remember that in order to translate our RNA sequence, we have to look at the codons. We have to start with the start codon, which is AUG, otherwise known as methionine. And then you continuously read the sequence of 3 in order to get your amino acid sequence until you get to a stop codon. We have 3 stop codons. Here's one of the stop codons. And then in the end, we have our polypeptide chain. We have that amino acid sequence, which in turn turns into a protein, which are very, very important. Now we want to look at what if there's some sort of mistake in our DNA. How is that going to affect our final amino acid sequence? And that's what we're really focusing on for today. So what is a mutation? The basic definition of that is a change in the DNA's nucleotide sequence. So something happens to that sequence which will then result in a different amino acid. Potentially, we're going to look at the different types of mutations. And now we want to be mindful that there are very, very harmful mutations, and this is going to result in a change in our DNA sequence and results in a protein that just does not work, or it can work in a really harmful way. So it really has a negative effect on the individual. However, there are some helpful mutations. And we kind of forget about that because we only hear about our harmful mutations, but there are definitely very helpful mutations. So this is actually when the protein works better due to the mutation, and that can result in evolution. We do want to be mindful, though, that it depends on the environment. So if the environment changes and we also have a change in maybe a protein that allows that individual to survive in that particular environment, that could then be a result or lead to evolution. And we'll talk about evolution a lot more in our next unit, which is very, very cool and very interesting. And there can also be neutral mutations where there is a slight mistake in the DNA sequence, but in the end, the protein is exactly the same. So this is a situation in which the protein is the same and there's no change in our protein. So that's just a neutral change. So what are some causes of mutations? Let's take a look. So there are a couple different things that can happen. There could be a mistake that occurs during DNA replication. So we have to remember that we constantly need to replicate our DNA because we are constantly creating more cells, and all of our cells need the DNA. So there could have been a mistake there. Doesn't really happen too often. Inheritance is another way where we can have mutations where an individual inherits a genetic mutation from their parents. Or we have a couple other things here in environmental issues, maybe a lot of exposure to UV radiation, exposure to certain carcinogens like pollutants and smoke. And I have down here this nice image, I think, that kind of breaks up several carcinogens that are all around us. So if we're looking at home and environment, pesticides definitely could have extreme issues where we're kind of talking about mutations, aerosols, UV rays, just going to the beach and not wearing any sunscreen for hours and hours is not safe. And you know that. Certain foods, like putting a lot of preservatives in foods, it's not good. It is much better to eat foods in the whole form. So eating like the fruits and vegetables that have not been processed are better than buying something like I don't know, in a package that has a lot of preservatives in it. Certain lifestyle choices like tobacco chewing, I don't know, but body piercings, I guess. Tar from smoking is another thing. Industry, certain types of paint. Viruses can also cause mutations to our DNA. So here are a few ways in which our DNA can become mutated. Now we're going to get into our mutations, look at the different kinds. I don't have these like a definition right here, but I want you to follow along, and in your notes you can kind of pause and define what each of these mutations are. So the first thing we want to look at is our normal sequence. So if we're given our mRNA sequence, which is up here, and this is our normal gene, nothing's wrong with it, and these are the proteins that we make based on our mRNA sequence. So here's our amino acid sequence. Okay, because now we want to look at how the amino acid sequence is affected. Nucleotide substitution is the first mutation we'll look at. So here we're going along, okay, that looks the same, right? Amino acid is the same, amino acid is the same, oh, same, but now we have a difference. Okay, so what exactly happened here? So for the substitution, we're noticing that before we had a guanine nucleotide, and now we have an adenine nucleotide. Because of that substitution, right here, we now have a different amino acid. And so a nucleotide substitution will lead to just one different amino acid, and all the rest would be the same, right? Everything else is like the same for that one. Now we want to look at a nucleotide deletion. When you delete something from the code, we start to see that there are a lot more issues that can arise. So for instance, we deleted uracil from our mRNA. Why is that such a big issue? Well, now because we deleted that, everything gets shifted in one direction, and it changes everything that comes afterwards. So before we have met and lyse, right, which are the same as our original, but now we have a different amino acid here. We have another different amino acid and another different amino acid, and it's because the whole sequence shifted. It shifted, and now we have that these three are a codon, and now these three are a codon, and now these three are a codon. And so this one, only one codon changed, right? Only one codon changed from the original. Now we have 3 codons that changed because of a deletion. Insertion is a very similar idea where we are inserting another nucleotide, and that will affect everything after that insertion. So we are changing the different codons, which then result in different amino acids. So what we notice is that a substitution, yeah, it does result in one amino acid change, which could have dramatic effects, right? It definitely could. But deletion and insertion, we can plainly see that there is definitely more kind of going on and more changes that occur. So let's just look at a few names of our mutations. We have a nonsense mutation. A nonsense mutation is actually when a codon changes into a stop codon. And so that actually cuts the whole process short. And now we're not going to have our long amino acid sequence like we did before. So now it just maybe cuts it in half, and we don't get to finish translation. A missense mutation is when one amino acid is basically swapped with another amino acid. So we can kind of see that down here. If this is our original strand with no mutation, and this is the amino acid we make, lyse, a nonsense is now we're just switching. A missense is just when we're swapping the amino acids due to a change in the sequence. Then a silent mutation is going to be where a nucleotide does change, but the amino acid stays the same. So for instance here, if this was our DNA TTC, a silent mutation might result in a TTT, but the amino acids in the end are exactly the same. So silent mutation, nothing happens, the protein is still going to be exactly the same. So it's due to the redundant code. Here's just a quick example of a missense mutation. This is with sickle cell anemia. So this is actually where we have, right here there's going to be a mistake. So this is our normal sequence. We have GAG. For a sickle cell anemia individual, they actually have a GTA. And because of that, that results in a different mRNA sequence and it results in a different amino acid. It's only one difference. That one letter is what causes somebody to have sickle cell anemia and has this a different amino acid, which then results in a hemoglobin molecule that is different shape. And we have our red blood cell is also a different shape. Because of that, oxygen has a really big issue traveling throughout the body. And so people with sickle cell anemia really have issues with transporting oxygen. They feel very, very tired. It's very painful for them. So this is just an example of a mutation, of a missense mutation. And this is the same thing, showing that same idea for sickle cell anemia, that one change results in a different amino acid in somebody that has sickle cell. All right, so this part, I'm going to ask you to pause the video and we're going to try to answer these. If we have the original strand is the cat is black and white with stripes, this is the mutated strand, so the cat is pink and white with stripes. Can you determine what type of mutation this is? And I want you to go through all of these as best you can and try to figure that out, and then we're going to go over it together. All right, so you're going to pause the video, do that based on what we just talked about, and then we'll go over it. All right, so here we have on the screen, I wrote this out for us. So the original strand is here, the cat is black and white with stripes. The mutated strand is saying that the cat is pink and white with stripes. So the answer to this is a missense mutation. The reason for that is because a missense mutation is when we're exchanging one amino acid for another amino acid. So this is the original, black and white, but now in our mutated form, it's going to be swapped with pink. So that is an example of a missense. So these are kind of representing our amino acids, and I'm representing it in a different way to try to express that idea to you. All right, so let's go on to our next example. All right, so now we have our next one, and we're seeing that our mutated strand is saying that the cat is black and white with stripes. There are no changes that have occurred here. So we're going to call this a silent mutation because there has been no change in the amino acid sequence. So we're calling this a silent mutation because the proteins look exactly the same. All right, next one. All right, and here's our third example. I made a slight mistake on the PowerPoint. There should be a D right here, which this is correct, but on the PowerPoint I'm missing that D. Just wanted to point that out. But what this is showing now is our sentence now looks completely different and it looks a bit crazy. So what has happened is now we've inserted this A, right? There was only one A before and now there's two A's there. So this is showing an insertion mutation. So we're inserting a letter which would represent a nucleotide, and that affects the rest of the sentence. And so now everything had to be shifted this way. Everything had to be shifted, and now the words don't make sense. And that's the same exact thing that will happen for a protein. When you have an insertion or a substitution, you're inserting something or you're deleting something, that results in everything else being affected by it. So a protein, everything else afterwards would be affected by it, and it messes everything up. And so that's what's happening for this example. The whole sentence doesn't make sense, and the same thing would happen for a protein. All right, so here's our next example. And this is an example of a deletion mutation. So what we'll notice is that before we had a K, and now we don't have a K. So the K was deleted from our sentence, and now we have to shift everything over. So the A will move over here, and then the N moves here, the D will move here, the W moves over here. So everything is being shifted over, and that results in, once again, our sentence doesn't really make sense anymore. And that's the same thing that would happen for a protein. Everything gets messed up after that either deletion point or the insertion point. Causes a lot of differences, a lot of changes to our final sentence or our final protein. All right, and then here's our last example where we have the cat is. and then nothing else afterwards. And so what this is representing is that there was a stop codon that was now being coded for, which it wasn't there originally. So because of this stop codon, this is a nonsense mutation, because of the stop codon, the rest of the sentence is unable to be completed. And now with the same idea for a protein. If a stop codon is in the middle of a sequence, then nothing else is going to code afterwards. So I hope those were helpful. It's just an analogy using sentences to help us kind of understand the different types of mutations. Okay. So this is what you're going to be doing for the remainder of today. You're going to complete the Google Forms on mutations. It's not a quiz, it's just to practice mutations. And then after that, I want you to work on the study guide for Monday's quiz. And the Monday's quiz will be a Google form, and it will count as a quiz, and it will be on protein synthesis and mutations. And there's a cute little picture of baby Yoda. This is kind of the look I get from a lot of students when your teacher tells you what to do, but you weren't listening, so you just stand there like, I hope you guys aren't feeling like that, but I don't know. All right, so I hope that was helpful. Please let me know if you have any questions, and have a nice day. All right, see ya.